We should be ready. Good. So I'm just going to give you a few words on bacterial genome sequencing just to set the scene for today's bioinformatics practical. So let's just start with a bit of audience participation. How big do you think the average bacterial genome is? Anyone want to throw us a number in terms of base pairs? 1 to 5 million is reasonable, yeah. So I think probably the average is about 2 million, but yeah. I think the largest ones might be up around the 9 million. And the smallest ones, well, it depends what you call a bacterium. The mitochondrial genome is only 13 or 15 kb, isn't it? But, okay, and how do bacterial genomes differ from the human genome sequence? Any salient features? Apart from being much smaller. Nobody's awake yet, are they? So you can say? Yeah, they only have one chromosome. They're usually circular, yeah. Not always, but usually. And the other thing, the key point is that most of the DNA in a bacterial genome actually is coding for proteins and represents genes, about 85%, I think, typically. Whereas in the human genome, about 1% of your genome or less is coding for proteins, because you've got large numbers of introns that take up huge amounts of space. And then what can you learn from a bacteria? Well, we'll go on to that as we go into the talk. So it's just to remind you the general features of genome. So microbial genomes, you might call them what you see is what you get genomes. It's very clear. You just look at the DNA sequence and you can see the protein coding genes very easily. You don't have to do too much analysis to get to them. And the G density is high. I put over 90%. I thought it was about 85%. But yeah, they're very short intergenic regions, very little repetitive or non-coding DNA, and introns are extremely rare. So it used to be thought there were no introns in bacteria, but about 30 years ago it was discovered that you can get introns, particularly in ribosomal genes, but they are extremely rare. And whereas in our human genomes, got very large genomes, very low gene density, and only about 25% of the genome is actually in genes, but in those genes, large parts of the sequence are taken up by introns. And the protein coding genes in a bacterium, because you get this what you see is what you get, they are 3 times longer in terms of numbers than the protein sequence, because it is just a direct relationship between the DNA, the codons, and the proteins you make. Whereas in the human genome, we have all these genes that are spanning massive distances, tens of kilobases, and they also you get splicing out of the introns, but you can get differential splicing, so you can get a variety of different transcripts all coming from the same gene. Whereas in bacteria, we have these operons where you get transcripts that reflect the operon. You may have several genes in a row, I think typically 3, 4, 5 sort of genes in a row. And the promoter regions are actually very tightly linked to the transcript. They're very close to it. Whereas in the human genome, they could be more distant. Now, I prepared this slide a little while ago, and I've put down there fewer non-coding RNAs. It's probably true that there are fewer non-coding RNAs, but actually it is clear now that bacteria do have non-coding RNAs, and this is quite a hot topic, actually discovering the non-coding RNAs and working out what they do. And the people who are devotees of non-coding RNAs will tell us that the protein genes are boring, and it's the non-coding RNAs that actually rule the cell. So that's an interesting point for the future. So as someone pointed out earlier, mostly we've got single circular chromosome, always DNA. Some species have linear chromosomes, and a few species have 2 chromosomes. and some even have a mixture of circular and linear. Many bacteria have plasmids, independent replicons that can replicate separately from the chromosome. They may have a copy number that is different from the chromosome, so there may be many more copies of the plasmid than there is of the chromosome. In some cases, they can jump into the chromosome, integrate into the chromosome, and sometimes that can be stable, sometimes it can be unstable. And they often carry what you might consider non-essential genes to core growth under all conditions, but they provide an adaptive advantage under certain conditions. So they effectively become part of the accessory genome of the cell. So when we're doing sequencing of bacterial genomes, as we've pointed out before, we start with the whole chromosome, but when we're doing Illumina sequencing at least, we shear that down and do size selection and add adapters and then sequence. So we're not actually directly sequencing the bacterial chromosome as a single molecule. We're ripping it into lots of pieces and then putting it back together. And then we have to assemble those sequences back to create the original sequence we started with. So here you can see that we can look at that green sequence there, and we can see that overlaps substantially with the red sequence there, so we could put those two together and make a contiguous piece of sequence out of them. And then we can see the red sequence there overlaps with the blue sequence there, and we can make a contiguous sequence out of them. What's the problem with the purple sequence? Anyone see any? Why is that an issue? It's what? Yeah, it's a repetitive sequence, so there are two repeats of that sequence. And the problem is that if you've got these short reads that actually are shorter than the repeat, you cannot reliably work out where they belong, whether they belong at one end or the other end of that molecule. And that means that you just effectively have to break your contigs at those repeats because you have no way of knowing what fits on either side of it. So that's the problem of sequence assembly, that it depends on the read length, and if your read length is relatively limited, you may have difficulties. Now, it depends. Some bacteria have more repetitive DNA than others. Typically, the problem comes with the ribosomal RNA operons, which they can be multiple copies. So E. coli, for example, has 7 copies of those in the cell. And so trying to work out the order of the fragments around those is a problem when you're doing sequence assembly. And as I mentioned yesterday, what that means is that when we do short read sequencing, we end up effectively with a draft genome that may be in dozens of contigs rather than being a completely contiguous genome. And this is just showing you the same kind of thing, that you end up with these contigs, and you can have gaps where you don't have any sequence for a particular region. The way you get around that is just by doing deeper and deeper coverage. So if you sequence a shotgun library, you don't get absolutely equal coverage of the whole chromosome. Just by chance, some parts of the chromosome you'll have more coverage and some parts you'll have less coverage. But you overcome that by just saying, well, we'll go 30-fold on average, which means that even the worst parts of the chromosome, you might, I don't know what it would be, 10-fold or 15-fold coverage, which is enough to be unambiguous about things. But the problem is if you get a physical gap where you've got bits of sequence that you can't actually clone or you can't actually amplify. This is less of a problem. This used to be more of a problem when we were making libraries in E. coli, because there would be some sequences that were toxic to E. coli and wouldn't come out in the plasmid library. They wouldn't be able to be replicated. I think that probably if you're dealing with extremes of GC or AT, you might still get issues with amplification and so forth. But so what the other thing that's worth saying here is that people you can put contigs together into scaffolds where you don't actually have the sequence that bridges the gap to turn 2 contigs into 1 contig, but you have other information that allows you to orientate them. You can then put them into scaffolds. So once you've got a genome, to make sense of it, you've got all these A, C's, T's, and G's. And one of my colleagues in the clinical lab, she thought that what we did was we just looked at the A, C's, T's, and G's, the whole list of it, and we could just, as humans, just read the sequence and make sense of it. And I had to point out, no, we're not like human computers. We have to go through and use computers to work out what is actually going on in that sequence, what it all means. So it's the addition of this information that we might call the metadata about the sequence that actually provides information that's usable by humans instead of just this list of A, C's, T's, and G's. So one of the chief tasks, early tasks of annotation, is to predict what are the genes, the protein coding genes. And we sometimes use the jargon term of CDS for coding sequences here to describe that. And this is a much easier task in bacteria than it is in humans, but there is still an issue. If you look just for open reading frames, which was the original way of doing this, you sometimes do get open reading frames that overlap, and only one of them is the real one that's coding a protein, and the other one is an antisense auth that isn't actually doing anything. And so there are more sophisticated methods now of actually predicting the coding sequences. Then the other major tool that we use is we use homology and homology searches. So what we do is we'll look for proteins in the database, or even sometimes DNA sequences in the databases that we can confidently predict are homologous, which means that they share a common ancestor, and because they have a common ancestor, we can start to make functional predictions as to what that gene is doing, what that protein is doing. In addition, there are a whole number of other features that we can annotate as well, and the amount of effort that goes into this might vary from one project to another, but you can see them there, the ribosomal RNAs, the transfer RNAs, trying to identify promoter sequences, non-coding RNAs, that sort of stuff. You can locate also, when you look at the bacterial genome, you can locate the origin of replication because of there's a sort of bias in the way in which nucleotides are used, and so that's a relatively easy thing to do. And people just count up the number of bases, the number of genes, the G plus C percentage of the genome when they're writing up these genome papers as general properties of the genome. One of the problems, as I said, with short read sequencing is you don't get assembled contiguous genomes. But if your interest is, say, in genomic epidemiology and you want to analyze an outbreak and you want to work out what's the relationship between these strains in front of you, and they're all fairly closely related, what you tend to do there, instead of bothering with the assembly and the annotation or whatever, you can just map the reads, the short reads, against a reference genome. So this is an old slide. In fact, this is when we used to have even smaller reads than we do now. This mapping process is like assembling a jigsaw, but instead of doing it de novo, you're using the lid of the picture on the lid. You know what your reference genome looks like, and you're just matching your reads up against that reference genome and assuming that everything that you want to know is in that reference genome. So if there are extra things in the strains that you're looking at that aren't in the reference genome, you won't uncover that information. It will be opaque to you. They won't get mapped, those reads. And similarly, if there are things in the reference genome that aren't in your strains, there'd be parts of the reference genome where none of your reads map. So there may have been a deletion, for example, in the strains you're looking at, and then you'll just see, oh, that part of the genome's not represented. And so this is the sort of picture, again, you're stacking up your reads, depends whether you've got single end or paired end reads, and aligning them against a reference sequence and seeing where they match. And then what you can do is where they differ from the reference genome, you can call out single nucleotide polymorphisms, or single nucleotide variants, as they're called, that can be informative when you're trying to analyze the epidemiology. So calling out these SNPs, some people purists tend to call them single nucleotide variants now, but the term SNP is very commonly used as well. But what this allows you to do, it allows you to do one of two things. So if you're looking at, say, the emergence of resistance, say you've got a TB strain that becomes resistant after treatment in the patient, you can then look at the changes in the genes responsible for that resistance, biologically significant changes, the result of natural selection going on within the patient under treatment. And that can be informative in terms of understanding the mechanisms of resistance and so forth. But it's not useful for working out the relationships between strains, because this is very constrained by natural selection, and you'll get the same mutations occurring in different lineages because of the same selective pressure. So if you want to reconstruct the family relationships, the phylogenetic relationships between strains, then you have to instead look for neutral changes. So what happens in a genome is that they're just copying mistakes in genes. As you can see there, in that alignment there, there's a G, it's been turned into a C. And most of these changes don't have any biological function. So if you think about the way the genetic code works, the third position in the codon often doesn't make any difference. So if you've got GG, you can have GGG will be glycine, but so will GGC will be glycine. And so it doesn't make any difference to the protein that's made. And even if you do make a difference to the amino acids, often the way the genetic code works is that the changes mean you just change to another amino acid that's fairly similar. So you might change from a glycine to an alanine or something like that. Those changes really don't have any biological significance, but they're very informative when we want to reconstruct the pathways that evolution has taken and the relationships between strains. And that's is now a very common activity among people sequencing genomes. And that particular tree there, it's a slightly unusual way of showing a tree. It's a phylogenetic tree, but it's been squashed into a circle. And this shows a relationship between various isolates of Staph aureus that come here from the Gambia that Madike did as part of his PhD. And he looked at isolates from monkeys, from the monkey part, from Bigelow, and also from humans, and showed that some of the monkeys were carrying strains they'd acquired from humans using this kind of approach. But this sort of approach can be used very, very widely to, for example, analyze a hospital outbreak, to track the spread of organisms in the community. And in the UK now, for tuberculosis, all the strains are being genome sequenced and they're being classified, and they can look at the relationships in a city like Birmingham and say, oh, that patient and that patient are very closely related. There must be some kind of epidemiological link, what's going on. Similarly, they're now sequencing all the salmonellas in the UK, well, in England and Wales at least. And so they can look at, so there's an outbreak in a Chinese restaurant in London. Oh, look, those strains are very, very similar to strains coming out of a Chinese restaurant in Newcastle, and it helps them reconstruct what's happening. So I've been around long enough that I can remember when all this started. I mean, it was very exciting when people first started sequencing bacterial genomes and using them in this kind of detective way of tracking down what's gone on. And the pioneering study, I guess, was with the so-called Amerithrax incident, where someone deliberately released anthrax spores into the American postal system. And by sequencing the genomes of that derived from the material recovered from the postal system and a strain stock from a laboratory, they were able to show that those anthrax spores came from a particular American lab, and it was an American homegrown terrorist, if you like, that was doing the damage. In fact, Paul Keim, who's a co-author on there, he told me that early on they didn't even use genome sequencing; they just used various VNTR approaches, and he said that by showing that the strain that caused that outbreak was just an American strain and a common lab strain, he was able to stop the Bush administration from invading Iraq by quite a few months, because Bush, when they saw the Amerithrax incident, they, oh, this is Saddam Hussein, they wanted to invade, but they held back. Unfortunately, George Bush had this obsession with sorting out Iraq, and he did go in eventually, but that was an interesting example where you can look at these genome analyses and it can influence things. Another highly political instance was the origins of the Haitian cholera outbreak. So there was an outbreak in cholera, and there was this suggestion that it might have been the UN peacekeepers had gone in after earthquake and trying to keep peace. And that was proven to be the case, that basically the cholera strain that was causing the outbreak in Haiti was very closely related to strains that were coming from, initially it was like the Indian subcontinent, but I think eventually in the subsequent study they even tied it down to saying these were Nepalese strains coming from, brought in by Nepalese peacekeepers. And that was, quite controversial, because although the UN were putting these people in there with the best intentions, people in Haiti, could be quite cross at the fact that the UN had actually caused a lot of damage by bringing in cholera. And then finally that one there, probable zoonotic leprosy in the southern United States. As you know, the United States is what we might classify as a shithole country, where the people do very strange things. And in the southern United States, people actually eat roadkill. And does anyone know what other animal apart from humans, what other animals, I should say, apart from humans, can carry leprosy? No, well, the nine-banded armadillo is a host in the United States. It's used in the lab. It was used in the lab to sequence the leprosy genome, because you can grow up large amounts of leprosy bacillus in the armadillo. But it actually got out into the wild and infected armadillos in the US. And what they showed with this study was that they showed that it got into the armadillos from humans. It wasn't native to America before Columbus or anything. It was taken into the Americas and then somehow got into the armadillos. And so the population structure of the leprosy in the armadillo is very, very restricted. But then they could show that there were some individuals who caught it back from the armadillos by skinning them and eating them. Strange things that people do. In fact, more recently from genome sequencing, it's been weirdest thing that I ever come across was that it's discovered that leprosy exists in the United Kingdom and in Ireland in squirrels. And it's been living in squirrels under our nose. We didn't even know it was there for hundreds of years. And so, you keep discovering new things. I'm starting to run out of time here. I mean, when you sequence genomes, you can start to reconstruct the relationships. You can look at clouds of diversity. You can look at how things fall together in an outbreak, how things are evolving in terms of their virulence properties or in terms of their resistance genes. And as I'm running out of time, I won't go through this in detail, but these are the sort of things you can do if you're looking at an outbreak. You can confirm the existence of an outbreak. You can start to define what is a case in your outbreak by looking at the genome. So, for example, in early days of sequencing TB genomes, they said, well, we're going to say that they're related if they have 10 SNPs or fewer between isolates. And that was a good rule of thumb to say, yeah, these are part of an outbreak. These are people who are, where the infection, acquiring infection from the same source or from each other. You can collect data, generate hypotheses, do analysis and hypothesis testing. You can check that your control measures are working, and you can also communicate with other labs. So if you can share the genome sequence between one context, say in England and Wales, between someone working in London and someone working in Cardiff, then it makes a very easy way to say, yeah, these are the same thing, they're the same, it's the same outbreak and so forth. And there are methods for doing this now internationally, for example, within the EU, in the US. Um, Again, I'm not going to go through this in detail, but we can generate sequences. We handle the data. There are issues and challenges there in terms of, that's why we're having this workshop, so that you can address these challenges. How do you analyze these data sets? How do you make sense of it all? And how do you perform epidemiological analyses? And we get, with whole genome sequencing, we're getting the highest possible resolution between different strains. We can say these strains are identical. These two isolates are identical. There is absolutely no question that there's some epidemiological link here. Whereas these are slightly different, still very closely related, but they're slightly different, and you can start to build up epidemiological investigations that way. Just one last slide here to say that there are other things that people do with high throughput sequencing on bacteria, apart from just sequencing genomes and doing epidemiology. One hot topic at the moment is doing RNA-seq, where you can look at which genes are actually expressed in a bacterium under different conditions by harvesting all the RNA, turning it into cDNA, and sequencing that. And this allows people to get a much more nuanced view of the genes that are actually in a bacterium, predict the genes, predict the operons, work out which transcripts are where, and look for non-coding RNAs as well that aren't actually related to production of proteins. And so this is another hot topic that people are using this for. There are challenges ahead. When you're sequencing genomes, genotype and phenotype don't necessarily correlate in a straightforward way. There are some, for example, if you're looking at antibiotic resistances, there are some cases where it's very, very clear. We have a lot of prior knowledge, and we can say that particular SNP in that particular location predicts ciprofloxacin resistance or rifampicin resistance or whatever. But there are other cases where it's not quite so clear, and that's a challenge. One area that's a growing topic is that when we sequence bacterial genomes and we pick a colony from a sample from a patient, we're making the assumption that that patient is colonized with a homogeneous population from that particular bacterium, but that may not be the case. It may be that the patient has got multiple strains of Staph aureus or multiple strains of TB. And by just picking one colony and analysing that, you might be making erroneous conclusions or misleading conclusions. I mean, this has been a problem in the past, say with sensitivity testing for TB, in that you pick the colony that's sensitive and say, oh, the patient's all right, but it turns out that maybe 7 out of the 10 colonies are sensitive, but 3 out of the 10 are resistant. Sometimes indistinguishable really does mean identical. So if you've got identical sequences from 10 individuals, you can't work out the relationship between those 10 individuals because there's no more resolution to be had. This is a problem for slowly growing, slowly mutating organisms. TB, for example, a bit, but certainly with Mycobacterium leprae, which grows very slowly and changes very little, it would be impossible to say, be absolutely confident that you could reconstruct who's given what to whom. Another issue is that in clinical microbiology, it's a very conservative discipline. So how do we move these technologies out from research environments in the ivory tower to the real world? This slide's, I've had this slide for several years. I'd have to say that I am pleasantly surprised at the way, at least in the UK, we have actually, the discipline has moved forward. And our reference laboratories are now using genome sequencing, whole genome sequencing. It probably is some time before this moves out into the peripheral labs in the district general hospitals. But it is quite heartening, the fact that the reference laboratories are now sequencing stuff. And obviously, if we're trying to communicate the results with clinicians at the other end and say, well, what does it all mean? They have to somehow become conversant with what we're talking about. And when I'm in my more florid moods, I can say, yeah, we'll just get rid of the whole of clinical microbiology and replace it all with sequencing. And all those people that like to look at plates and pick colonies, they're just going to be consigned to the baggage of history. And the great march forward is towards sequencing. But in my more kind of temperate moods, you have to remember that sometimes old technologies survive alongside new technologies, and the new technologies don't completely replace them. We still listen to the radio, even though we also have the television, and you think, the television's far more informative and a superior thing. So I don't know where we're going to be in terms of sequencing genomes and how far that will go if we come back in 5 years or 10 years or 20 years. I'm not going to say for definite that we'll get rid of agar plates in my lifetime, but we're certainly, sequencing will come in and it will make a difference, is making a difference.